I guess I'll jump into the pool first, and all I can say is, wow. It would have been much easier for me, I'm Ira Herman, to present some of our work on how pericyte chemomechanics teaches endothelial cells to regrow after being growth arrested. We've heard about modeling in animals and in vitro. We've talked about pathogenesis and human disease. We've talked about how oxidative stress might be a player, whether hemodynamics and biomechanics play important roles, how the microenvironment influences cellular dynamics. Professor Aird started us off by teaching us that not all endothelial cells are actually created equal, and that organ-specific differences might be actually dependent or regulated by the cells that are born in those organ systems and the way in which those tissues come together to give rise to the organ-specific functionality that we see. And then, in addition, we have also, and I think importantly, and maybe this is a good point to jump off from, talked about how vascular cells not only talk to one another and how they modify matrix, and for example, in glomerulonephritis, or whether we're talking about a disease in the brain or whichever organ system, it may be about the way in which the microenvironment, the non-vascular cells, actually talk to the vascular cells that shape the phenomena, whether they be physiologic or pathophysiologic. In the case of, for example, aquaporin-borne channels in astrocytic foot processes that are uniquely organized around special blood vessel features that penetrate the brain parenchyma to help take CSF out of the organ while we sleep. I don't sleep enough. Maybe that's my problem. So if there's not something here for you to talk about, to ask about, we can start asking questions or talking amongst ourselves. But we challenge you, the audience, to perhaps go ahead. Please.
This one. Yeah. Sorry, Tritana Galileo. So I wanted you to think a little bit about something else. And we talked here about the small vessel cell interactions, but I think there is a theme that I want you to think about. And the vascular is not really in its own. I mean, it interacts on one side with pericytes and the tissue-specific cells to form organ-specific properties, but also interacts with immune cells. And I think one of the themes that I want you to think about here is the interaction of the immune cells with the vasculature. And I think Dorian McGovern talk tomorrow is going to bring back that discussion as well. But you know, this came from the first talk, thinking about lupus. And even though the lupus discussion was more on the kidney, it actually also has a lot of neurological effects. So I want you to kind of, you know, maybe some of the questions that are going to be thought about really bring back that, you know, thinking in terms of an underlying theme that brings immune cell CNS interactions. The second talk by Gary really described very nicely this particular disease, but, you know, the CNS has sort of an immune privilege property, but there's a lot of interaction of the immune cells with the CNS, and that becomes very prominent in diseases of the CNS, such as MS or this disease. And in particular, I think also some of the stuff that Nika talked about, for a long time we thought that the brain doesn't have a lymphatic system, but yet this particular lymphatic really brings in a new concept of how we think about brain physiology. So there may be really an underlying theme in all the organs that I want us to discuss, because I think part of this workshop is really to, you know, think of a unifying theme of the microvasculature. So, you know, maybe we can sort of streamline the discussion in some of these 3 points.
Okay. Open to questions from the floor now.
So I have a question. I guess it's really to make it. Okay. So you mentioned that proteins come out of the perivascular space with pulsatile flow. But what drives this? I mean, is this vasomotion? Is this, you thinking of cardiac motion of the vessels? if it's cardiac motion, they all seem to go up and down together. So that means you need valves. So where are the valves in the perivascular space?
Weller have described valve-like structures in the Wecken-Robin space in human patients. He does very, very good EM studies of valve-like structures in around human arteries. So I would also say that, of course, that the exit of protein from plasma to CSF or to interstitial fluid in brain is, of course, much smaller than it is peripheral tissue because of the blood-brain barrier. But we do have leakage of proteins into the interstitial fluid, and especially, and this is what I try to talk about, is that it looked like all cytosolic protein, even though they don't have a secretory sequence, are released to the interstitial space and can be found both by microdialysis by David Holtzman in the interstitial space, but also in the cerebrospinal fluid.
And what's the pulsation?
Also, we just published a study that showed that pulsatility, at least in part, is responsible. So you can drive these fluxes of cerebrospinal fluid up and down by changing the pulsatility, but it's not the whole story.
But that's vasomotion?
It's vasomotion, yeah. And whether that's just a cardiac pulsatility or whether it's a very slow motion you have described, we don't know.
John Lederer, University of Maryland. I don't understand what is special about sleep that permits, during that period of time, the cleansing of bad proteins. Can you describe this a little better?
It's a good dream. No, we actually showed, and we did quite a bit of studies on it, that there is no. Basically, sleep is a default of the brain. Arousal is modulated by all the neuromodulators, norepinephrine, orexin, and so on. And what the neuromodulators do is not to drive different level of lymphatic fluxes to sleep and awake by pulsatility, but what happens is that the interstitial space in the brain parenchyma expands 65% when we go to sleep.
So why is that?
Our cells shrink as a function of being exposed to norepinephrine in particular.
So what is contracting and what is expanding? I mean, is all that worked out?
It's underway, but what I know is that there's papers in press in human brain showing, using diffusion-weighted images, that water movement is restricted the less you sleep. So if you sleep-deprived patient, you have less water movement in them than during the sleeping state.
So Mike, what's the role of the neuron in all of this?
Yeah.
So I am not talking about whether it's astrocyte or neurons shrinking or expanding. I don't know, because we don't have the optical resolution to explore that. What I know is interstitial space changes in volume.
Maria Luisa Sequeira Lopez, Charlottesville, Virginia. Continuing with the same theme, do you see the same changes during all sleep stages, or the rapid eye movement stage?
That's a really good question. We haven't explored it in detail yet because mice have only very short-lasting, REM episodes, and we only have 2 shots of the traces. So we are exploring it in larger animals that have more solid REM sleep to see whether there's a difference or not in that, but we don't know yet.
And another question is, what would happen during exercise? Because your mouse is resting when you are studying it. So what do you speculate that will happen?
It goes even further down. I would expect that. Yeah.
Hi, Dave Antonet, Michigan. Dr. Lopez, I wanted to ask you a question. What do you think it would be like to expand the kind of system that you're working with so it is more readily available and maybe even commercially available in the scientific community? And maybe associated with that, what do you think of the systems that are already out there, like the cartridge systems and such? And some of your thoughts on that.
So for the second question, I can't really answer because I haven't used them. We either use parallel plate chambers that we can coat with endothelial cells, or we use this system. We are trying actually to expand this to make it more available because it is kind of finicky. There was a company that was interested, Ibidi was interested in this, and they still are, I think, but we don't really have the whole intellectual property issue was a problem. But yeah, I think what we need to do is we need to make it easier to use, and I guess that the complex one, if you need to do anything complicated, then you're probably going to have to, do a lot of work. But there's a lot of simple things that you could use with a simpler model.
Do you see differences depending upon whether or not you use, for example, venous endothelium from umbilical vein, or whether you use capillary-derived cells?
Well, Ying has probably done that more than we've done that in our lab, and she does see differences, although what we also see is we see something that Dr. Ayer talked about this morning. We see a lot of plasticity depending on what you do to the cells. For example, I didn't have a lot of time to show this, but the megakaryocytes completely change the endothelial phenotype, absolutely completely change it, and make it like they wanted to make, as if it were in the bone marrow.
Just back to the sleep conversation. I was wondering if you've looked into patients that have LVAD implants, so they don't have pulsatile flow.
We haven't, but we have reduced pulsatility, and it reduces the perivascular influx of cerebrospinal fluid. So we haven't looked at it.
I wonder if we could get back to the first talk and just talk a little bit about how antibody-mediated injury influences the glomerular adaptive responses. And I'm particularly interested in knowing whether or not the modifications that you make using transgenic approaches to the macrophages, or leukocytes in this case, actually are feeding directly to the endothelium, or how does the mesangial cell talk to the endothelium to give rise to the glomerulonephritic disorder that you see? In the PAS localization that you showed, the mesangial matrix was remarkably hypertrophied. Mm-hmm. And so I couldn't help but think that the mural cell talking to the endothelium was a way in which there was sort of crosstalk coming back from the parenchyma. Is that true or not?
Yes, so mesangial cells are sort of akin to smooth muscle cells and parasites and other vasculature. We don't work so much on mesangial cells, but certainly they contribute significantly to glomerulonephritis. But just getting back to just a point on immune cell endothelial cell crosstalk, definitely there's something there, but what I wanted to actually also bring up is that there's something about the target organ susceptibility factors, and this is probably in the CNS and anywhere else, that dictates whether that immune cell will actually even react to the deposited immune complexes. So in our case, we had to actually knock out a receptor on MAC1, which we think regulates Fc gamma 2A, in order to see the injury. But there may be some other endothelial factors, some mesangial factors, epithelial cell factors that also give rise to that ability of the immune cell to sort of see the immune complexes and make them pathogenic. And that also gets back to some model systems that when we talk about, we use naive mice, for example, in all of our studies, and we all do this with C57 Black 6 in our facilities. But many of the diseases we talk about, including lupus, have a lot of comorbidities associated with them. It may now be part of that disease susceptibility, target organ disease susceptibility factors. And so we should also think about mouse models that maybe are slightly more sophisticated and can replicate what happens in humans.
For in the case of diabetes.
Exactly. Yeah. Right.
So I had a question related to that, just keeping the theme of the immune cell endothelial interactions. In your knockouts, have you looked at other disease models where neutrophils play a role? For example, there's very clear evidence of neutrophils implicated in stroke progression. There's also role in MS. Mm-hmm. So have you looked in those knockouts to see if there are profound, changes, just like what you described in glomerulonephritis?
So in our MAC1 knockouts, which we generated a long time ago, MAC1 is normally thought to be pro-inflammatory. Right. MAC1 is a CD18 integrin, is supposed to be important in leukocyte recruitment. But then since then, it's been really appreciated that it can be immunomodulatory. And we think in the context of immune complexes and Fc gamma receptors, actually dampens down. And interestingly, there's a SNP very highly associated with lupus in MAC1, and that SNP leads to reduction in MAC1 function. So the model that we had developed actually may have some relevance, so that in this case, when you actually take away MAC1 function, it seems to hype up the immune system, versus in other sterile inflammation like with TNF, etc., MAC1 may actually be pro-inflammatory. So there's a yin and yang of many of these molecules.
Do you know which of the ligands for MAC1 is involved?
Oh, we'd love to know that. We think it may be complement or ICAM. We don't know. We'd have to do that experiment.
So one more question from the floor.
Yes, Jose, I was just wondering, endothelial cells make ADAMTS13. Can you just comment on its regulated release and whether you've looked at, for example, knockdown, whether you can make those strings even larger in cells that don't secrete?
That's a very good point. Well, I actually don't know anything about its regulated release. I think it's constitutively released. I don't think there's a lot of it in most endothelial cells. Most of it comes from hepatic stellate cells. Probably, so we have some experiments, and I think these might address this. We haven't knocked it down, but we put high concentrations of antibodies that will neutralize it in there. And the extent of strand elongation and thickening is even more rapid. So I think there is something that's coming out of the endothelial cells.
One more short question.
Short question for Dr. Niedergaard. Comments on effects of fluctuations of serum sodium and water fluxes related to sodium levels, how that might impact your glymphatic system.
That's really interesting, because the glymphatic and the cell volume changes is, of course, all about iron homeostasis. So maybe we can talk about it, but I think it's extremely important, and I think one of the things we are working at now is actually to show that the interstitial iron composition differ in sleep and awake, and that can again basically drive arousal, because if you just increase potassium by 0.5 millimolar, you change the whole excitability of cortex. So it's not just volume changes.
Okay, an applause to the discussants and chairs and speakers.